Good morning. My name is Monica Mir. I'm going to share my screen. Okay.
Okay, perfect. We can see your screen. You can proceed with the presentation.
Yeah, but let me see if I can move the slides because it was not happening before. And I think it's not happening now. Okay, now? Yeah? Can you
Yes,
see
we can.
the
Yes,
Okay,
yes.
finally.
Perfect. Yeah, go
Perfect.
ahead.
Okay, so my name is Monica Mir. I'm from the Institute for Bioengineering of Catalonia, and I'm going to present our work in electrochemical implantable sensors for in situ monitoring on the body. And to do a brief introduction of the research group where I'm working. It's the nanobioengineering group at IBEC. So basically, the group is divided in three main research areas. One area in micro and nanoenvironments for biomolecular interactions, where we are studying different 3D co-culture of cells and its interaction with nanometric structures. Also, there is the area of microfluidics, where we are developing different kind of lab-on-a-chip systems and the study of bio interactions in microfluidic systems. And also the area of biosensors and point-of-care devices that we have developed different kind of point-of-care systems for companies and basically for clinical diagnosis. And the combination of the three areas, so the 3D co-culture of cells, the microfluidics, and the monitoring with biosensors give us also the opportunity to develop organ-on-a-chip in vitro models that are complex systems that integrate all these areas. But for today, I'm going to introduce my work in electrochemical implantable sensors for in situ monitoring of ischemia in the body. So basically, ischemia is a lack of perfusion of blood inside the body, and then the lack of oxygen. And there are different areas of medical application for this ischemia. For example, detection of hemorrhages, the flat post-surgery, so the perfect anastomosis between two flats. The compartmental syndrome, that is a trauma that blocked the flow of blood in a certain area of the body. Also, fetal growth restriction, that is restricted the pass of blood through the umbilical cord, and also myocardial ischemia. So there is a clear unmet need, medical needs for solving this problem. That is basically the point that when in a tissue there is a lack of oxygen or a lack of blood perfusion, there is around 5 days to detect with the current methods. And in fact, the tissue is degraded if there is ischemia in 1 or 2 days. So when we detect the problem, the tissue is already degraded that we need to replace this tissue. So we need a fast and early monitoring of ischemia for medical diagnosis. The current methods that are commercially available for this detection is, for example, the Korg-Schwarz Doppler, that is basically a sensor that is put it surrounding the vessel, so it's quite high risk for introducing the sensor, and is quite expensive because you need surgery for adapting the sensor. Also, there is the Stricker needle, that is a needle that you can insert at any part of the body and is an indirect measurement because it's basically measuring pressure. That is not directly, is indirectly related with the lack of oxygen. Also, there is the possibility of microdialysis, that is the extraction of blood sample or tissue sample for detecting the lack of oxygen. But we had have discussions with different key opinion leaders in different hospitals here in Barcelona, and they commented that the way of they are currently using for detecting ischemia in the tissue is basically the clinical observation. So when they observe a change of the color of the tissue, a change of the temperature on the tissue, they can detect the problem of ischemia, but then it's too late to recover the tissue. So in order that you have an idea about the impact of this problem, I put you some numbers in different applications. As you have seen, there are many other medical applications, this is just numbers from US market, and for example in reparative surgery, but just in plastic surgery. So it's very little market comparing with the whole surgery or reparative surgery that is down in the whole planet. So it's around 200,000 patients, and it's very costly to make a second surgery for the reparation. Also, compartmental syndrome has around of 3 million patients per year just in United States. So it's a quite market required for the solve of this problem. The technology that we propose is an implantable sensor that will be placed at any part of the body, injected into the tissue, that will be externally connected from the external part of the body, and then with a kind of nanometric wire of electrodes that basically will be connected wireless with a laptop for monitoring the evolution of ischemia in the tissue. So basically, we are trying to make a continuous monitoring of ischemia with a multi-analyte sensor. So we are going to detect more than one analyte in a miniaturized needle of around 500 micrometers. It's going to be implantable, flexible, that permit deep analysis of the problem, is not risky, and easy to use, and has low cost. So basically, we compare with the current methodologies that we have right now. The technology that we propose has no risk, has a continuous monitoring, permit deep flap monitoring. So basically, the lack of blood in the tissue generate a lack of oxygen, since the blood is basically transporting a part of many other things, also oxygen. Then the cell, when it has this lack of oxygen, the first thing that we can detect is obviously the concentration of oxygen, but also that generates a kind of cascade of events, like the decrease of the ATP in the cell, that makes that the pumping of sodium and potassium is reduced, then is changed the concentration of sodium and potassium, and then can be detected also through pH, through potassium sensors. And also the lack of oxygen make that it generated an anaerobic respiration of the cell, then producing acid lactic, and then decreasing the pH in the tissue. So we are also able to detect it with a pH sensor. So basically, we are trying to combine two different analytes in order to have more information, so oxygen and pH, with this micrometric wire of electrodes in an array format with electronics incorporated into the system. So the first sensor that we have developed is the pH sensor, that is based in a polymer, that is the polypyrrole. That is a polymer that has the attraction of protons. So we are able to detect this attraction of protons on the polymer due to a change of charges, because are positively ions, the protons, and then we are able to detect with a difference of voltage. Here you can see we use, in fact, electropolymerization on the electrode surface of this polypyrrole, and you can see here some pictures of the modification of this surface, and also the calibration curve of these sensors with different also interferent molecules. Here the calibration curve with a good nurse response, and also the response after 18 months of working with the sensors, that is not changing so much the slope of the response of the sensor. Also, we developed the oxygen sensor that is based in the detection of the reduction of oxygen. So basically, it's this reaction. So basically, the reduction of oxygen to water, and we are able to detect it with a cyclic volt amperometry at a voltage of minus 0.8. That is basically this peak. Here you can see the response of different oxygen concentrations. And also, if we modify, that's a platinum electrode, with Nafion that has better response. Here we check different membranes for the detection of oxygen, and the one that gives us better answer with standard oxygen molecules and also with blood samples is the platinum with nafion membranes. And here you can see the repeatability of the response of these sensors with different concentration of oxygen. Also, we made some tests of the sterilization of this kind of wire electrodes, because basically we need to introduce inside the body, so we need to be sure that we are able to sterilize this kind of sensors and that the sensors are not highly affected by this sterilization. So sterilization that works better for us was the UV, ultraviolet, that just reduce 4 millivolts of the response of these sensors. Here you can see also a long-term analysis of the sensor of pH. And once we optimize with in vitro samples our sensors with controlled solutions of pH and potassium, then we are ready for going a step forward, so to test our sensors in vivo conditions. So we developed 2 different models of ischemia for testing the oxygen and pH sensors implanted inside animals. So we start first with small animals with a rabbit model for analyzing ischemia in muscular tissue that could be related with medical applications of analysis of hemorrhages, compartmental syndrome, or floods post-surgery. And also we develop a model for fetal monitoring in sheep model that is correlated with fetal growth restriction. So I'm starting with the rabbit model. In the rabbit model, we start with the anesthesia of the animal and to control the signs, the vital signs of the animal. And we introduce the sensors inside the muscular tissue of the rabbit in the leg. So it's this kind of wire sensors that are flexible and can be introduced with a catheter at any part of the body. Then we control the breathing of the animal with this equipment. Basically, we can increase or reduce the amount of oxygen that is breathing the animal. And then also we are extracting continuously blood, not continuously, just when we are in the stages of normoxia and hypoxia, we're extracting blood samples in order to characterize the state of the animal with a standard equipment. And also we are monitoring continuously the evolution of the animal and to analyze the different stages of normoxia and hypoxia. Here you can see the results with the pH sensor in ex vivo. That means measuring the blood samples that we extracted from the animal. Here are the values of normoxia, and we can see a significant difference with hypoxia conditions. Here you can see the measured values of hypoxia and normoxia. And also we are trying to recover to normoxia the animal, but considering as that we are measuring the formation of pH, the pH is not removed so fast from the blood, and we are not able to detect the recovery. Similar results were obtained when we are measuring in the tissue, directly in the muscle of the animal, with normoxia, so we can see significant differences between normoxia and hypoxia stages with these results. And then we move to the different sensor, that is the oxygen sensor, where we can see more clear differences because it's a direct measurement of the lack of oxygen. So the pH was a consequence of this lack of oxygen and was not so clear, but in the case of oxygen, we have high clear difference between the 2 stages, and we were able also to see the recovery because basically we are measuring in blood the injection and the block of oxygen, so as a direct measurement, so we can clearly see the recovery of this oxygen concentration in blood. That is not so clear in tissue, so we need to see a diffusion of this oxygen for see the recovery, but also we can see very clear differences between normoxia and hypoxia with these oxygen sensors implanted into the animal tissue. Also in the fetal model, with the sheep model, we go ahead for testing our sensors. So the idea was to have a model to analyze with human fetus growth restriction. So it's a problem that makes that the fetus died in before the born of the fetus. So basically, the idea is to inject a sensor in the leg of the muscle of the fetus for doing a monitoring of the evolution of the fetus. So we had made this model in a fetus of a sheep. We start also with the anesthesia of the animal and the control of the signs, the vital signs of the mother and also of the fetus inside the sheep. Then we insert the sensors inside the muscle of the animal, of the fetus of the animal. Also these micrometric wires. And then in this case, instead of controlling the breathing of the animal, what we are controlling is the pass of blood through the umbilical cord. So what we have done is put a kind of ring that restricts the flow of blood through the umbilical cord. So in this case, we can control the flowing of blood that is coming into the fetus. In the same way as before, we was extracting blood samples in normoxia and hypoxia stages, and we was continuously monitoring the evolution of the fetus. Here you can see the results that we had obtained in the fetus model with intramuscular, so with the sensor injected inside the leg of the fetus, and intravascular, that in this case we did it with the injection of the sensor inside the vessel of the fetus. So that's the advantage of having so micrometric size of the sensors. Here you can see that was clear differences between the normoxia and hypoxia again, also problems for detecting the recovery, but except in the case of the blood detection that we are able to detect recovery. In this case, we are able to differentiate between 50% of occlusion of the umbilical cord and 100% of occlusion, and we're able to see a trend on the response. That's for the pH sensors, and for the oxygen sensors, we are also getting good results in the intramuscular analysis, also in the ex vivo with the blood, and we can see significant differences between the normoxia and hypoxia stages. So if you want to get more information about the experimentation that we have done, the research that we have done in this kind of implantable sensors for ischemia detection, you have different publications. Here you have the information, and also a brief introduction of the projects that we are working currently in implantable sensors. So we are working currently with a European project that is called 4Lab, where we are implanting directly in the vessel sensors that are completely wireless. So it's a powerless consumption electrochemical sensors, able to detect different kind of analytes, pH, potassium, but also proteins with an aptamer beacon system that we are developing. So it would be a quite powerful technology. And also in the area of advanced in vitro models, we are mimicking the functionality of a part of the brain in a chip. So basically, we are mimicking a co-culture of the vessel of the brain with a neuronal culture in close contact with the endothelial co-culture with pericytes and astrocytes, with sensors integrated into the microfluidic system for analyzing neuronal activity and permeability of the barrier, the blood-brain barrier. So we are currently working with this so complex technology right now. And well, you are welcome to have questions about this technology and for collaborations. Here is my email address. So if you have any question, any doubt, we can discuss it. Do you have any question? Okay, thank you.